Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall look at two different programmed cell death. One is ferroptosis and the second is cuproptosis. Ferroptosis was reported in 2012, and this is a programmed cell death that is related to several important diseases, including cancer, various neurodegenerative diseases, cardiovascular disease, as well as liver diseases. Now, in cases where there is excess iron in the body, what we call iron overload, this increases the chances of ferroptosis. And you see iron overload in several situations. For example, the genetic disease hereditary hemochromatosis, where the patients absorb too much iron. There is iron overload that can happen because of frequent blood transfusions or taking high doses of iron supplements. So whatever the reason, the presence of excess iron could result in what we call the Fenton reaction, something we talked about in video 8.8. So iron, the Fe2 plus form, can react with hydrogen peroxide. And as we talked about in earlier videos, hydrogen peroxide is produced by several different reactions in the human body. And this reaction, this Fenton reaction, can produce a very powerful and dangerous free radical called the hydroxyl radical. So iron can lead to the production of this hydroxyl radical. Now, one of the effects of hydroxyl radical is that it can react with polyunsaturated fatty acids very easily to convert these fatty acids into lipid peroxides. Okay, so the PUFAs become PUFA peroxides. So in this reaction, the hydroxyl radical will react with the PUFA, polyunsaturated fatty acid, forming a PUFA radical. Now this PUFA radical can then react with oxygen to form PUFA peroxide radical. Then the PUFA peroxide radical can react with another PUFA, forming once again the PUFA radical. This PUFA radical now can react with more oxygen to give you PUFA peroxide, just like what we saw here. And then the PUFA peroxide can react with another PUFA, just like what we have here, to give you more PUFA radicals. So if this reaction is not stopped, the PUFAs gradually will be converted into PUFA peroxides or lipid peroxides. Now, PUFAs are very important components of several different membranes, including the plasma membrane, as well as the membranes of the mitochondria and other organelles such as lysosomes and ER. So when these PUFAs get damaged, get converted to peroxides, that is going to lead to severe membrane damage, not only the plasma membrane, but membranes of various organelles in the cell. So we can see all kinds of problems here when all these membranes are being damaged. For example, if the plasma membrane is ruptured or becomes more permeable due to the conversion of these PUFAs to PUFA peroxides, or sodium and water can enter the cell, as we saw several times before in other programmed cell deaths. That can lead to swelling of the cell as well as organelle, further membrane damage. Then all kinds of chemicals, cellular contents can come out of the cell. Some of these chemicals can act as danger-associated molecular patterns, and they can trigger inflammation, and eventually, due to all this membrane damage, the cell will die. In this case, due to iron overload, and therefore we call this cell death ferroptosis. Now, there are two very important factors that can affect the chances of the cell dying by ferroptosis. if there is a lack of this very important enzyme, glutathione peroxidase GPX4, or if there is insufficient reduced glutathione, so either lack of this enzyme or low levels of GSH, these two factors can significantly increase the chances of a cell dying by ferroptosis. Now in video 8.8, recall that we talked about this enzyme called glutathione peroxidase. We saw how this enzyme, glutathione peroxidase, one of the reactions it can catalyze is breaking down hydrogen peroxide into something very safe like water. But to do this reaction, it requires reduced glutathione. The reduced glutathione becomes converted to oxidized glutathione. So to reduce the oxidized glutathione back to reduced glutathione so that this reaction can keep on happening, we need another enzyme called glutathione reductase. So glutathione reductase will convert oxidized glutathione back into reduced glutathione, and this reduced glutathione can then be used by the enzyme glutathione peroxidase. So besides converting hydrogen peroxide to water, glutathione peroxidase also will convert the lipid peroxides into lipid alcohols. So the PUFA peroxides will become PUFA alcohols. And once again, we see the requirement for reduced glutathione. This can only happen if there's enough reduced glutathione. And this reduced glutathione comes with the help of the enzyme glutathione reductase, which converts the oxidized form into the reduced form. So therefore, it's not surprising that ferroptosis, the chances of a cell dying by ferroptosis increases tremendously, either if this enzyme is not present to stop this formation of lipid peroxides, or if there is not enough glutathione available for this enzyme to carry out its function. A few words about the morphology of cells that die by ferroptosis. There is a lot of mitochondrial changes. There's atrophy of the mitochondria. The mitochondrial membrane becomes very thick. There is disruption of the mitochondrial membrane. You see decreased mitochondrial ridges, or what we call crista. The nucleus looks normal. There is no chromatin condensation as what we see in apoptosis, and also there is no formation of membrane blebs as what we saw in apoptosis. Now, the other programmed cell that we'll talk about is cuproptosis, and this was reported fairly recently in 2022. And once again, not surprisingly, this is also related to several diseases, including cancer, neurodegenerative diseases, cardiovascular, and liver disease, as well as several other diseases. In this case, there is a copper overload, and that can happen by a genetic disease called Wilson's disease, where the body cannot get rid of excess copper. It can happen due to environmental reasons. For example, the water that we drink could be contaminated with copper, using copper cookware or copper glasses to drink water, excess copper supplements, and of course copper poisoning, since copper is used a lot in for making all kinds of copper pipes and so on. So one way by which copper can cause cuproptosis is shown here. The Cu2 plus can enter and cross the plasma membrane without any transport protein if it's chelated to certain chemicals, what we call ionophores. So when the copper is chelated to certain chemicals, the whole complex can enter the cytoplasm and eventually be transported into the mitochondria. Now in the mitochondria, we have this reaction where several proteins, mitochondrial proteins, are lipolylated. Basically, lipoic acid is attached to the protein. Lipoic acid is attached to the protein. Proteins such as pyruvate dehydrogenase, complex components, components of alpha-ketoglutarate dehydrogenase, these proteins are lipolylated. Now, this reaction is carried out by enzymes such as lipoyl synthase with the help of this enzyme ferrodoxin 1 or FDX1. Now, FDX1 can reduce this copper 2 plus into copper 1 plus. So besides being involved in helping in lipolation of proteins, FDX1 is also involved in steroid as well as heme A synthesis, metabolism of bile acids, and vitamins A and D metabolism. So when this FDX1 converts Cu2 plus to Cu plus, now Cu plus accumulates, and this Cu plus can interact with these lipolylated proteins and cause aggregation of these lipolylated proteins. So, for example, the dihydrolipoamide acetyltransferase or DL80, which is a component of PDH complex. So when these proteins become aggregated, these enzymes such as your PDH complex and your alpha-ketoglutarate dehydrogenase, they do not work anymore. Another thing Cu plus does, it destabilizes iron-sulfur clusters. Now recall that iron-sulfur clusters are very predominant in the electron transport chain. So this destabilization of iron-sulfur clusters by Cu plus is going to impair the function of the electron transport chain, while all these lipolylated proteins that are aggregated, that's going to impair the pyruvate dehydrogenase complex, the Krebs cycle, components of the Krebs cycle, and that's going to lead to impaired mitochondrial metabolism. So these effects eventually will, of course, cause very severe cell stress, and the cell will die due to this cell stress. The mitochondria especially will be very severely affected. There will be a shrinkage of the mitochondria and disruption of the mitochondrial membrane, and all of that is going to cause cell death. Now, another way the copper can cause cuproptosis is by actual reduction of the copper into Cu plus, and this is done by proteins in the plasma membrane. These are enzymes that help the uptake of Cu plus, metalloreductase enzymes. So these enzymes will reduce Cu2 plus to Cu plus, and then the Cu plus is transported into the cytoplasm. Now, if there is excess Cu plus can also bind and inhibit the ubiquitin proteasome system. So it inhibits the proteasome and interferes with ubiquitination. So with this system being inhibited, remember this system is important for destroying excess proteins. So if this system is inhibited, proteins are not destroyed. So more and more damaged proteins will accumulate in the cell. And this accumulation of proteins is going to lead to cell stress and eventually cell death. Since this cell death is due to copper, this would be cuproptosis. So this is a little bit about ferroptosis and cuproptosis. In the next video, we shall look at another pathway, programmed cell death, and that's called oxyoptosis. So until the next video, once again, thank you very much for watching and supporting this channel. Have a wonderful day, and we'll see you in the next video.